This phase right here is interphase. So if I ask you what phase is this, you're going to say interphase. Do not do what the lab supplement says. We're going to call this interphase. If I ask you what is the name of this structure, you're going to say it is the cell membrane. If I ask you what is this area, you're going to say the cytoplasm. If I ask you identify the structure in blue, you can either put down nucleus or nuclear envelope. It doesn't matter to me. If I ask you what is this green structure, you're going to say nucleolus. If I ask you what are these disorganized type of chromosomes, I usually won't use the word chromosomes. I'll say identify these structures in purple, and the answer would be chromatids. So the disorganized are going to be chromatids. This right here in black and this right here in black is going to be the centrioles, and now we're going to move on to the next one. So again, that's interphase. This right here is going to be prophase. So during prophase, we now have these things that look like butterflies. They're going to be called chromatids. We have these things in black here, which are going to be centrioles. The white structure here are going to be the spindle fibers, cell membrane, cytoplasm. So this is prophase. This is also prophase because we know that the centrioles are continuing to move apart. So as we go back and we look at both of them together in the same frame, we can notice that the centrioles are starting to move apart here, and they're finishing their moving apart here. That's what's defining both of these as being prophase. So this is prophase. Again, the structures in white here are going to be the spindle fibers, and these again are going to be called chromatids. As we move to the next one, this is now going to be metaphase. We know it's metaphase because the chromatids line up on what we call the metaphase plate, which is found in the middle. Remember, letter M, metaphase, middle M. The structures in red are going to be your spindle fibers. The black are going to be your centrioles. The structure here, if I ask you about this, is the cytoplasm. Here is the cell membrane. And again, this is metaphase. So we go to the next phase, and we can see that we still have the chromatids, we have the spindle fibers, and we can see that the chromatids are starting to separate. So we can see that they're no longer that butterfly that it was before. So if we pan back a little bit, we're going to notice that these are sort of have like 1, 2, 3, 4 connected. Now we're seeing that 1, 2 are on one side and 1, 2 are on the other side. So they're starting to separate, and that's what's making this anaphase. Again, we have the centrioles. This right here is going to be the cytoplasm, cell membrane, and again, this is anaphase. This is also going to be anaphase, and if we pan back a little bit, we're going to see that both of these are anaphase. This is just the beginning of anaphase, and this is toward the end of anaphase. Now, don't worry about telling me middle, late, early, anything like that. Just call this anaphase, and you're going to call this anaphase. If I ask you about the phase. So again, the question has to be about the phase. If I ask you what is the name of these white structures here, you're going to again call them spindle fibers. The purple is going to be chromatids. The red are going to be spindle fibers. The black are centrioles. The area here is the cytoplasm. This area right here is the cell membrane. As we go to the next phase, this is the final phase of mitosis, and this is telophase. We can see we have the reemergence of the nuclear envelope. We see the pinching in. That's also an indication of telophase. Those two are two pretty good indicators of that we're dealing with telophase. These are going to be your chromosomes. These in green are going to be the nucleolus. These in black are going to be your centrioles. This is the cytoplasm. This is the cell membrane. Okay, and then once we're done with telophase, again, this is telophase, we then get back to two of the same cells, and these cells again are going to be interphase. So this part, as we pan back to look at the entire board as a whole, we can see that this is interphase. These two cells are interphase. We went from one cell to two cells, interphase. So we have interphase, prophase, we have prophase, metaphase, the chromosomes in the middle, metaphase plate, anaphase, the chromosomes are starting to separate. This is also anaphase because the chromosomes are continuing to separate. And then telophase, where we have the nucleus re-emerging and the pinching in of the cell membrane. And then again, we went from one cell to two cells. So again, interphase, prophase, metaphase, anaphase, telophase, interphase.